Hello everyone, welcome to our channel. So today we are going to discuss plant breeding and genetic, which is most important topic. So before going to start the video, we have to know that what are the meaning of genetic. So let's start it. So, meaning of genetic. So, genetic means it is a process in which transfer of gene or character from parent to offspring or sibling. This process is known as genetic. So, inheritance is also similar to genetic, in which it is also transformation or transmission of character from parent to offspring. This process is known as inheritance. And now we are going to discuss plant breeding. So what are plant breeding? So plant breeding is a technique which improve the crop fertility. We know that from ancient time that is not facilitated, that time technology is not developed, then crop is damaged and totally diseased. So now new technology, biotechnology are released and new experiment are doing day by day. So in this method or technique which improve the crop fertility. So crop fertility, which means crop produce a new crop which is better than previous one, and disease resistance. There is no insecticide, pesticides are present and harm to that crop. So this process which we are study in plant breeding, which help in agriculture purpose for making better crop for human being as well as animal also. What are the steps of plant breeding? So first step is collection of variability. So first of all, we have taken one plant and another plant. So that first plant is taken germ plasma, and second plant so also collect a plasma, and going to hybridization process from both plant, and selection and testing and superior combinate, they are superior or not, they are good or not, they are check everything, then they are combined and testing and make a new variability. So this process is known as process of plant breeding, which are used in plant. Suppose that there is one plant, there is second plant. In the process we have to choose one plant and second plant. So first plant, first plant should not should a good superior plant, as well as second plant also. That's why they are combined. When they combine and become a, with the help of hybridization process, and make a new plant, recombinant plant. So this process is known as plant breeding technique. So as we all know that there is an example. So in this example is a brassica aloracea. This is a common wild mustard. So this example taken from mustard plant. We have experiment and choose this plant. We make with the help of plant breeding method or technique. We make cabbage, cauliflower, broccoli and kale and many more plants with the help of mustard. This is a wild mustard. So as we all know that, who is the father of green revolution? Green revolution, what are the meaning of green revolution? So green revolution means growing more green tree in our agriculture, which are given by Dr. N.E. Broland in 1960 and M.S. Swaminathan. This is the architect of green revolution in India. So M.S. Swaminathan is architect of green revolution. He is stand for green revolution in India. Then there our country is also improving and thoughting and developed our crop fertility with the help of green revolution. And there are many tools or techniques comes and to improve the crop fertility. So what are the role of genetic? So what are the role of genetic in plant breeding? So genetic is the help in plant breeding is most important. So with the help of genetic, which we could not improve the crop fertility without genetic process. We are going to study in biotechnology process, courses. So we are going to discuss, there are many various type, but we are going to discuss only three types. So their first one is formation of hybrid. So first of all, form hybrid. So they are in this process make a hybrid and making old plant to new plant with the help of genetic and technology. So next so in this process we can go for tissue culture. So tissue culture, it means make a plant. So make a plant. So suppose a plant and extracted a piece of leaf, and next is a take a seed from other seed or germ cell from other plant, make them leaf and that other seed to combine and provide isolated cells and give a medium condition to grow a plant. Then they are grow in agriculture purpose, which is this process is known as tissue culture, which help in biotechnology process. So next one is recombinant DNA technology. In this process what happen? So agriculture, it means gene and cell DNA or recombinant DNA, this process are isolated from and combine, and their process is going recombinant hybrid is come, then they are mixing into plant cell and they grow into new plants. So this process is helping plant breeding in which genetics help in plant breeding, which is very useful for our agriculture purpose. So next topic, cell, it is a structure and cell division. We are going to discuss cell and cell division. So as we all know that cell is a basic structural and function unit of every life, which is a plant or animal and human. So as we know that Robert Hooke saw the first time and coined the term cell. So he is also known as cytology cell. He is also known as cytology. And A.V. Liu Wenqiu first time saw and describe a live cell. He described a live cell. Robert Hooke saw them a cell, but A.V. Liu Wenqiu saw the live cell. Later, Robert Brown discovered the nucleus. So there are two types of cell: prokaryotic cell and eukaryotic cell. So prokaryotic cells are unicellular; they are present in unicellular organism, while eukaryotic cell, it is a present in multicellular organism. So eukaryotic cell is unicellular. They are shown only one cell, which 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 are only division of oneself and making itself cell division and make it same similar clone वगैरह. So there we are going to study in uh, prokaryotic cell. So eukaryotic cell, they are multicellular organism. They are multicellular organism means they have more than one cell and division of lots of cells and make a tissue. This process is known as eukaryotic cell. Mostly prokaryotic cell is study in plant, while eukaryotic cell is going to study in human being and other animals. Now we are going to discuss structural organization of cell. So as we all know that cell is the basic unit of life. Cell are divided into three types. There are three kinds of cell wall and plasma membrane. Second one is photoplasm and dytoplasm. So cell wall are present in found in plant, bacteria and fungi, and photoplasm are present only living organism, and third one is dytoplasm present in non-living. So we are going to discuss photoplasm. Photoplasm is a living, so there are two kinds of. First one is cytoplasm and nucleoplasm. So cytoplasm, it is we are going to discuss outside of the nucleus. Suppose that there is a nucleus, so we suppose there is cell, and in the center there is nucleus. So there are many organelles present inside of the cell, such as mitochondria, chloroplast, endoplasmic reticulum, Golgi bodies, ribosome, centrosome, mitochondria, and vacuole. So what are the role of mitochondria? It is also known as powerhouse of the cell, and it is a help in oxidative phosphorylation and generation of ATP, which is provided energy of every living organism. Chloroplast is a help in providing pigments of that organism. In their chloroplast, they are present plasticide, leukocyte, chromoplast, chloroplast. So next one is endoplasmic reticulum. There are two types of. They are smooth reticulum and rough reticulum, which depend upon other organism, but they are basically important types of endoplasmic reticulum are rough and smooth, which help in transport of substance such as protein, lipoprotein, and glycogen. And what are the function of Golgi bodies? So they are packaging and secretion of substance, and ribosome, which help in protein synthesis, as centrosome is give us to spindle structures during the cell division in animal cell. Microbodies, there are perosomes and glycosomes. Vacuoles is helps in transport the ion and material in plant cell, which contain in amebia, which we can show in amebia and food process. So now nucleoplasm is going to discuss inside of the cell, inside of the nucleus. This is nucleus, we are going to discuss in nucleus. So in the inside of the nucleus, there are presence of DNA, RNA, as well as protein. So now we are going to discuss cell division. So this cell division, as we all know that in single person, they have 23 number of chromosome, and what they make daughter cells. As we all know that a single cell is divided into two types, and they make identical daughter cell. So they are two types of phase. This is also known as amphases, which is divided into two phases. First one is mitosis, second one is meiosis. So first one is to show 2 n number of chromosome, while meiosis show n number of chromosome, which is dependent upon the various cell division method. So first one is interphase. So interphase, so we are going to discuss metaphase. So interphase, in which cell the centrosomes and chromosome are present, they are 2n. So their DNA is copied and chromosome paired. So their DNA copy, and the centrosome and DNA make them similar copies, and there it is going in photoplasm. So prophase steps, the chromosome pair up. Metaphase, the chromosome line is equator, the chromosome, they are going to equator position, and they make a meiotic spindle. And anaphase is a sister chromatids pulled apart, and telophase and cytokinesis, in which the cells are divided into two, they are divided into middle and make a two identical daughters. They are diploid in nature. They then this process is continuous, as well as they are going on. So now Now going to discuss meiosis. So meiosis are two types of meiosis. First one is meiosis first, second one is meiosis second. So they in a meiosis first there is interphase, same procedure we are going to discuss in meiosis. So their interphase are similar, centrosome are present in top portion, and the chromosome make their similar copies which shown in for prophase, and second metaphase, third step metaphase, the chromosomes are up and make them equatorial, and anaphase is a chromosome pulled apart, and telophase and gynophase cells are divided. While they are divided, then the meiosis second process is going steps. So prophase second, in which two daughter cells are divided itself, making same chromosome copies, and third anaphase is making same sister cells, and telophase and gynophase making they are make and break into middle portion and make a four daughter cell, and their number of cell is n. So this process we are showing in multicellular organism, which are eukaryotic organism. So now we are going to discuss principle of inheritance. So as we discuss, inheritance means transfer. It is a process which transfer the characters from one generation to next generation. So G. J. Mandel in 1966 give this law of inheritance. So he took a example of pea plant, and doing an experiment on pea plant. So what he do? So he take a tall plant, he take a parent, two tall plant and two dwarf plant. So tall plant are male, and dwarf plant is female. So they are both plant are different from each other, and they both plant are crossed pollination, and they are becoming all tall plant. So now they are self self cross, self pollination or self cross. So for second generation they show them that they are three talls and one dwarf, while further they are selfing, they show all tall, but three tall, one draft, three tall, one draft, three tall, and all draft. This is also so show 9:3:3:1. Now we are going to discuss a principal paired character, which is first law of dominance. It is stated that when a pair and alleles are together in hybrid, and them expressed in the following completely. So they are also taken examples of different plant. Take a gene and combine a cross pollination and give us a pink flower. This is red flower and this is pink flower, give us pink flower, while they are self pollination, they give us um, one one red flower, one pink flower, one pink flower, one white flower. So it means it ratio show 1:2:1. So this law is given by Mendel. So second law is a law of segregation. It means a separation of gametes from parent to offspring. So this process is known as segregation of genes. So in this process they are going to take an example. So they take a tall plant and dwarf plant, similar plants. So they are taken eggs and pollen grain, and they are self pollinated and give us 100% tall in first generation, while the segregation their gametes and pollen grain, they are self pollinated and give a second generation is 75% tall and 25% draft. So In third law, law of inheritance assortment. So in this process we have taken in dihybrid hybridization process, so in which taken two plants. So they are taken yellow plant and wrinkle plant. So they are going to self both are self cross pollinated and give us 100% round yellow, while the segregation of allele and segregation of allele, so both are give us egg cells and pollen grain, and they are further they are self pollinated and give us second generation is 3 round yellow, wrinkle yellow, 3 round yellow and 1 green yellow. So now, what are self-pollination and cross-pollination? Self-pollination occur in same plant in which transfer of pollen grain from anther to stigma on same flower or same flower. So this process on same plant, this process is known as self-pollination, while cross-pollination occur in two different plants. Same procedure are applied on this transfer of pollen grain from anther to stigma, but it is a condition cross pollination occur in two different plant, while self pollination occur in same plant and cross pollination is occur in different plant. Thank you for watching, and do not forget to subscribe and share with your friends. In this lecture you have any doubt, drop a comment on comment section. PDF is also available. You can contact me in the given link of Telegram.